Okay, this is going to be our review of mitosis, meiosis, the chromosomes. We'll go over all the anatomy of that, and we'll go over the cell cycle. So what should you do? You should take notes. Draw pictures. And write down questions. So I will try to keep this as brief as I can and keep it to what you absolutely need to know and what I've noticed that you're confused about. Okay, let's start with the basic structure of a chromosome. A chromosome is DNA when it's condensed. So normally DNA is all, whoopsie, there we go, all kind of stretched out. Okay, because we need to read the DNA. So this is a really poorly drawn double helix, all stretched out. And that's so we can read the information of a DNA. But during mitosis and meiosis, chromosomes condense during prophase so that we can easily move around chromosomes without the chromosomes ending up in the wrong place. So they don't look like this during mitosis and meiosis. So, oopsie, trying to erase my terrible drawings, but they're not going anywhere. Hello, disappear. Okay, I'll erase them the old school way. Okay, so what do the chromosomes look like when they're in their condensed form for mitosis and meiosis? So we're still talking about DNA, but in its condensed version. Okay. So first of all, what is that? I'm going to put a little line here for you to label what that would be and when that would be. So you might want to pause for a second, and I want to know what that is and when that is. I'm going to draw some more and put all the labels in and then explain. Okay. So now okay. So now what is it now that we have two halves, and when would that be? So what is this entire thing called? Okay, and now in the middle here, there's a structure that sort of temporarily holds these two together. And what is that called? I'll put a line here for that. And along chromosomes, we have different segments. So what are those? Okay, so we have a few things to label on the structure of a chromosome. So first of all, I wanted to know what 1/2 of this was and when that was. And let's see if I can do that without making this look like a mess. So, kind of want to know what 1/2 of this was. Okay, and then I wanted to know about the center that joins the two halves, and segments along a chromosome, and then I just gave away that term of what it's called, the entire thing. Okay, so now after you've paused and drawn this structure for yourself, let's do some labeling. So first of all, in G1 of the cell cycle, before chromosome has duplicated itself, okay, we have just the one side, okay, just this half, and I'm going to let me try to use the pointer for a second, which I think is this. Okay, so this here, okay, is what we would have during G1 before the S phase. So that is, once they're together, it's called a chromatid, but it's a chromosome all by itself before it is replicated. All right, and I want to get rid of that. Okay, there we go. So once they're together, of course, then it's a chromatid. Okay, so do some labeling here. Yep, think. Okay. Okay, so when it's together, they're each called chromatids, but before replication, it's an unduplicated chromosome. So, unduplicated chromosome . I hope I'm being clear there. So before G1, in G1, it's an unduplicated chromosome, that half. But then once it's together, they're sister chromatids. So then it becomes a chromatid. And then the entire thing is, of course, a chromosome. This middle red piece that holds them together, centromere. So segments along a chromosome, a segment of DNA is a gene. And we'll get more into that, but it's important just to notice that a gene is along the same place on each side. Okay, so this arrow was just pointing to the chromatid there. Okay, and that the half was the G1 unduplicated chromosome. So for cell cycles, if we compare the basically mitosis and meiosis. The somatic cells, remember somatic means body, so over here will be the mitosis cell cycle. Just to quickly go through it. I don't think we need a huge reminder on this one. So we have gap 1, we have the S phase, we have gap 2, and then we have mitosis and cytokinesis. Okay, so remember that in gap 1 we have growth. The cell does its normal cell things, and then in the S phase is when we have DNA replication. And in G2 we prepare for mitosis, and then by the end of cytokinesis we have 2 cells. And so, let me change colors here, everything from here to here is interphase. So most of the life of a cell, like your skin cells and stuff, are in interphase, and then for a short time they're dividing. And so you have one cell and it comes around, and then over here you have one cell and it divides into 2. Okay, and so just as a reminder too that your body cells have 46 chromosomes, okay? And so in G1 it's going to have 46 chromosomes. When it hits the S phase and the DNA replicates, it will have 92. And so in G2, it's going to have 92. And so over here, right at the beginning of mitosis, it will have 92, so that each resulting cell will have 46. So that the resulting cells here, let me turn on the laser pointer, the resulting cells here look identical to the beginning cell from over here. And we'll go through that when we talk more about mitosis. But I just wanted to remind you what this whole cell cycle looks like compared from mitosis and meiosis, because it's really the same thing, except that instead of mitosis in this position, we have meiosis. So over here in in meiosis, we have this stem cell that's just going to go through this phase, this cell cycle, this one time, and then become a sex cell, so stem cells become something else. And so it's going to go through meiosis 1 and 2, and then that last bit of cytokinesis is here. Okay, so in this case, got the screen. Okay, we're going to be starting with, well, and this is still all considered interphase also, okay? And so we're starting here with a cell that has 46 chromosomes, okay? And then when it comes to DNA replication, it goes to be 92, okay? And then when it goes through meiosis, it goes through 2 divisions. It's called a reduction division. So when it's beginning of mitosis, it has 92, and then it's going to go from 1 cell to 2 cells to 4. I can't fit them all in there, but we'll talk about it. And so those 4 cells each end up with having 23. Let me just draw them down here. So our beginning cell has 92, these have 46, and our gametes, the resulting cell of meiosis, have 23 chromosomes. So that way sperm has 23 and eggs have 23, and then when they get together they form 46. So the cell cycles of each for the stem cells and for body cells look kind of the same, except that they have meiosis here and mitosis here. The other big thing is that the somatic cells, this is happening all the time. Your skin cells are going through their cell cycle all the time and going through mitosis. Whereas stem cells becoming sperm is only happening in the testes, and it's a very unique process. And for females, it all happens inside the ovaries before girls are born. So that's completely different. It's not happening throughout the lifespan. So that's a huge difference between these 2 cell cycles. Okay, so for mitosis, to start with, big questions. What cells go through mitosis? Why do cells go through mitosis? And then how does the process of mitosis occur? So what are the phases and how do they go? We've been through this many times, and you should have that trick down of whatever you use to remember PMAT. As you know, I use Paul Makes Taco, and I've been using that a long time. Okay, so you should pause for a second to write down those big questions. Those are always on MCAS, and those will definitely be on your test to know what cells, why, and how. Okay, so what cells? Well, as we said, these are somatic cells. Your body cells go through mitosis, but there are 2 exceptions. After you're fully grown, your brain cells and your liver cells no longer go through mitosis. Why do cells go through mitosis? For growth and repair. So if you have damaged cells, they need to be replaced. And then the how is cell division. Prophase, metaphase, anaphase, telophase. Paul makes taco. And then over here, I drew a phase. Okay, what phase is this? Okay, and then how can you tell? So I know it's not an awesome drawing, but how can you tell what phase it is? Well, there's a few things to cue you in. For one, you might be able to tell that my poorly drawn nucleus is breaking down, and you should be able to also tell that these chromosomes are duplicated. What would these chromosomes look like unduplicated? So if you guess that this is prophase, that's correct. So let me just write that down here so that you know. Okay, it's prophase. Okay, so this cell has already gone through the S phase. So at this point, okay, these chromosomes have been duplicated. If this was a human cell, which it's not because we're only looking at a small number of chromosomes, obviously, but if this was a human cell, we would have 92 chromosomes instead of 46. So things are doubled. So we can see that things are doubled. And this is important because down here I have possible outcomes. So if you had to draw the outcome of mitosis, would you pick this as the outcome or this as the outcome? Knowing the outcome would be identical to the original cell, which outcome would you choose? Okay, so mitosis is the production of identical daughter cells. Now, this is not the original cell. This is a cell in prophase. So you have to back up your brain and think, what did this cell look like in G1? Because that would be what we'd want to have two identical daughter cells looking like. So what did this cell look like? Okay, the other way to figure it out is to go through metaphase, anaphase, and telophase. All right, so I'm going to quickly draw metaphase, anaphase, and telophase just to show you, to make sure that you know, although went through it a lot. Okay, there we go. All right, so I'm going to put my metaphase there, my anaphase there, and my telophase is going to look like that. And I've got a black chromosome lining up along the, oopsie, erase that one. Come on. Okay. Here's my black chromosome, metaphase, middle, lining up along the middle. There, and my light green one, which is supposed to be a little bit smaller. It's hard to control my my iPad pen here. Okay, and then anaphase. Oh, I should draw in my spindles here. Let me make my spindles kind of brownish. Okay. With my centriole there, okay, because we really need those spindles for, whoops, that was a little big, for pulling them apart for anaphase. Okay, so here's where you can really get a sense of what they would look like being pulled apart in anaphase. Okay. Oops. And you can kind of now start to guess what things would look like in the finished product if you couldn't see it right away. And I realize that this video is pretty long, but hopefully it has allowed you to review all the topics here and get everything studied for your test. Okay, and also in telophase, we start to have the beginnings of a nucleus forming. Okay, and then after this, we start to have the cleaving of the cytoplasm before it goes through cytokinesis. So to review what happens, in prophase, our chromosomes condense to become visible. The nucleus starts to break down. In metaphase, they line up along the middle. are spindles attached to the chromosomes to get ready to pull them apart. These spindles are attached to centrioles. Okay, those are called centrioles. And those are called spindles. Okay. And in anaphase here, the spindles pull the chromosomes apart. In telophase, okay, we have them beginning to form two new cells. The nuclear membrane begins to form around the chromosomes, and you can see that we have our chromosomes looking like they must have looked for the original cell, and our cytoplasm begins to cleave. Then we have cytokinesis occur, and we have our actual outcome of mitosis here. So this is the outcome of mitosis. And this one is incorrect. Okay, so that the original cell in G1 must have looked like this prior to the chromosomes being replicated in G2. So I hope that really helped clarify mitosis for you. Often we compare mitosis and meiosis, and as I've said, I don't always teach them together because they're so different, and the similarity is that the chromosomes are organized in such a way to make division possible. But MCAS likes you to compare them, and it's a common way to think these things through. So let's take a minute to just think about the similarities and differences. So I told you that my trick for remembering the difference is that mitosis is mito. So I remember that it's body cells. So this is body cells. And meiosis is sex cells. Okay, and for mitosis, we are making identical cells. Where I should probably put a line here so we keep things straight. Meiosis, we're making unique cells. Okay. They are both division. So this is cell division. This is cell division. This goes through PMAT one time. This goes through PMAT two times. The result here is 2 cells, and the result here is 4 cells. Okay, so hopefully that helps you keep some things straight. In humans, let's see, the cells, these 2 cells have 46 chromosomes, and these 4 cells have 23 chromosomes. The big thing is identical versus unique, because the whole point of meiosis is to make unique cells to have increase the variety in the species as an advantage for evolution. All right, so let's review meiosis, and this video will be all done. It's key. With meiosis, a really key question is, when do the chromosomes become unique? And how? Oh boy, that's nice writing, Mead. Okay. So this says, when do the chromosomes become unique? And I'm also asking how. Okay, so this is a super important point, as I said, because this is the big secret to sexual reproduction. So, there's one, and here's its friend. Okay, so when I draw these together like this, what's that called? Ba-ba-dum. What is that? That's a question. If you said homologous chromosomes, you're right. So this is after they've been replicated in the S phase, and when they line up together to be near each other to do crossing over, okay, this is a tetrad when they're all together. Tetrad. Okay, tetra means 4. Okay, so homologous chromosome, homo means same, so it means we have the same genes in the same location. So it's not going to work if I have, say, a whole bunch of genes for eye color here, and I had hair color over here. So let's say we have eye color here, and eye color here. Okay, so these genes are going to hang out near each other, okay, and then one is going to cross over the other and exchange genes. So I'm going to erase this one because it's going to cross over here. It's going to come over here, and they're going to exchange genes that are in the similar location that code for the same stuff. So say it's all genes for eye color, which is not just one gene, it's a whole bunch of genes that code for eye color. So they're going to cross over, literally crossing over in location and exchanging genes. So, okay, crossing over, and that's happening in prophase 1. So that's happening right away. The other thing is that, so this is one part. The other thing that happens is independent assortment. And I made a bunch of classes. Oh, that's not independent. It's late. I've been pushing it to get this done, and I'm a little tired. Independent assortment. Which means that okay, that this one, oops, this one, and this one could be lining up on either side. So this one could be on the right, and this one could be on the left, or they can switch places. And that's independent assortment. Lining up right or left. And it's not really right or left, but just it could be up and down or whatever. But for the sake of seeing it here, and that's why I put eyeballs on them so that they look like they're doing the cha-cha there. So you can imagine that they're dancing, that they're going to be lining up either way in whatever order they want to, kind of randomly, so that they're lining up independently. So we're talking about homologous chromosomes, chromosomes with the same genes, lining up whichever way happens, and then they cross over and exchange genes. So they line up randomly and exchange their genes, and that will eventually, when all of these guys end up splitting up, we'll end up with 4 cells that are totally unique because they'll have unique genes. Okay? So these 2 very important events that you must know happen in prophase 1. It doesn't happen again in prophase 2. It just happens right at the beginning of mitosis in prophase 1. Okay, so moving meiosis. Big picture is a good way to think about it. So we have our stem cell. It has 46 chromosomes. It goes through the S phase of its cell cycle where DNA replicates. So then it has 92 chromosomes. It goes through meiosis 1. So PMAT, prophase, metaphase, anaphase, telophase 1, and we get 2 cells, each with 46. Then each of those go through meiosis 2, PMAT again. And then we end up with our 23. And these are called gametes. These are not really our sex cells yet. They're gametes and they're going to develop further. They don't look like sperm yet because they're not sperm yet. They are going to be. So they have to develop. They don't look like eggs yet. They have to develop. And for every 4 gametes we get, just one of them becomes an egg. Eggs are a little bit more specialized. But this is the basic process. And so because we go from 92 all the way down to 23, meiosis is called, instead of just regular division like mitosis, it's called reduction division because we reduce in number. And that's sort of a nice to know, not a need to know. So it's called reduction division. So you don't have to know that, but that's a thing that kind of pops up from now and again on MCAS. But it's a reduction division because we go from 92 to 46 to 23. Because as we split them down and make more cells, we have less chromosomes in each. Okay? you have a lot of notes and drawings on, because we've spent much time on what happens in all the phases. Okay, so, but I just want to show you in one slide what the gametes look like, and just to go over zygote and fertilization. Okay, so our gametes are going to have, okay, a nucleus, and then a bunch of those single type chromosomes like we saw before, where it's those single lines. Okay, so like pre-replicated chromosomes we saw for mitosis cells. So this is a gamete. Okay. Okay, and it's haploid because it has half the chromosomes of a normal cell. Because it has, for humans, 23. Remember, all sexually reproducing organisms, flowering plants, other mammals, sexually reproducing insects, they're all going to go through this. So, okay, so we have our 23 chromosome sperm meets our 23 chromosome egg, and oopsie, and at that moment is called fertilization. And that creates a single cell of 46 called a zygote. That is the first initial cell created from fertilization. And it's unique because each of our 23 cells were unique, and so you are made of half the cells of your mother and half the cells of your father in that unique combination. And so that is it for meiosis, mitosis, and the cell cycle. Please bring any questions you have, and we'll go over them before our test. Good.